네, 안녕하세요. Today I'll be talking about how GLP-1 increases pre-ingestive satiation via hypothalamic circuits in mice and humans. So I think we all know that obesity is a big crisis around the world, and it's even considered an ever-growing pandemic. And the main reason for this is because of these palatable, fattening food that we can easily get access to everywhere around the world. And due to these food, the rate of obesity in almost all the country around the world has been increasing every year. So then the question comes to what is the cause of obesity? I think that many people in the past thought that genetics was the problem, but now more focus is coming into the metabolic features and the behavioral aspect of the causes of obesity. So this is the picture of a woman very fat and not moving, and due to the inactivity of uh, after we consume a lot of food, we don't use a lot of energy like before, so we get fat. And another aspect is that we just eat much more food. We generate a lot of eating behavior, and this overconsumption leads to obesity. Then what is the cause? Then where in the body does eating behavior generated? I think we can all agree that the brain is the core of our body that generates all of our behavior, and of course it also generates eating behavior too. Then where in the brain is this eating behavior generated? The hypothalamus has been known as the feeding center for many, many years. And so what the hypothalamus basically does is it integrates nutrients and metabolic demands, hormones, into the hypothalamus, and then it calculates the total calories in the body, and then it produces a hunger or satiety signal that that induces a feeding behavior, which then regulates the homeostasis. So then, so if so, in order to understand how the hypothalamus controls feeding behavior, we first have to understand the fundamentals of feeding behavior. So the feeding behavior is not a simple process. It's known to be divided into mainly three phases, and each phase has its unique characteristics and neuronal circuits. So when we become hungry, we start the food procurement phase, which is also known as the appetitive phase. And what we do here is we go out search for food because we're hungry. And then when we discover food, we grab it and we start consuming. So this leads us to the phase two, the food consumption phase. And this consummatory phase makes the food come into our gut, and it innervates our gut, and this gut signal sends a feedback signal to the brain, and this leads to a meal termination that 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 makes us stop eating. Then, of course, if this meal termination is not properly administered, then it will lead to a sustained consumption, which will lead to overconsumption, and that will lead to obesity. So in real, obesity patients report that they have problem controlling when to stop eating. So this means that we have to understand the fundamental process of meal termination in order to really control, uh, uh, in order to really get to know how to regulate obesity. So the meal termination is also divided into a few phases. The first phase is the satiation phase, which is the sensation of getting full after we start eating a meal. So after we start eating a meal, we start eating, and then when the food comes into the gut, we start to get full, and then we think like, oh, I'm full, then I need to stop eating. So that satiation feeling leads to a meal termination. And then what is satiety? Well, satiety happens after meal termination. So uh so it makes us feel like, okay, I've had enough food, I don't need to start eating again. So that prevents us from eating again in the meal initiation to start us going to a meal initiation phase again. But the satiation is not only divided into is also divided into to many more phases. So cognitive and sensory aspects of the satiation of our body also affect satiation, such as thinking of how much we'll eat in the future, looking at very oily food. So the sight, smell, and taste or touch of food, they affect satiation, and that leads to a meal that also affects our meal termination. So in our study, we tried to focus on this pre-ingestive satiation, which is a process of feeling satiated even before the food comes into the body. Then, so our body and all animals have this meal termination mechanism. Then how can we use this mechanism to control obesity? So in the past, the GLP-1 has been discovered in our body, and this sends satiation signals to our brain, which leads to a meal termination. So these GLP-1 drugs have been developed, um, ha analogs have been developed in the past, and after the launch of these GLP-1 drugs, there has been an explosion of the need for these drugs because of their phenomenal effect on body weight. So due to this incredible effect and the need for these drugs, the science journals have designated them as the breakthrough of the year in 2023. But despite all these wonderful things, the problem is that we don't understand the mechanism of the GLP-1 drugs. So we know they trigger insulin release from the pancreas, they block glucagon secretion, they slow stomach emptying, and they increase how full you feel after eating. But we don't know where in the brain it works or what kind of mechanism that I just explained it controls specifically. But the people who took the GLP-1 drugs in our lab, they said that even looking at food make them feel full, or they don't even want to see the food again. And then there was a recent study that showed that people who were treated with GLP-1 drugs, they had an increased of brain activity, and uh, they could they could see that using fMRI study. So we thought that the GLP-1 drugs could induce pre-ingestive satiation, and we wanted to investigate where in the brain this would be taking place. So we first thought we needed to look into the clinical aspect, if we could quantify pre-ingestive satiation in a clinical study, because there weren't a lot of studies that really looked into if we could looked into investigating pre-ingestive satiation in humans. So what we did was we recruited about 30 Korean obese people, and we defined them as obese if they had a BMI of over 28, or if they had a symptom related to obesity. And then we divided them into two groups. Then we let them do a task before or after they took a drug. And the task is here. So what they first do is they come and they do this baseline period where they come right away, and then they answer a questionnaire that asks them, how much do you feel full? So then we can get satiation scores from this question. Then we go on to the pre-oral period where we deliver the people with a plate full of 순살 BBQ chicken, and they can see it and they can smell it, but they're not allowed to put it in their mouth. And then we go on to the oral period where people can take the chicken and then they can put it in their mouth, they can bite it, they can taste it, but they're not allowed to swallow it. And then after that, we go on to the ingestive period where people can freely eat the food however they want. So the results we got is here. In the control groups, you can see that as people experience more food information, you can see that the satiation scores decrease. But in the GLP-1 groups, people who are treated with GLP-1 drugs, you can see that the satiation score increases as food information is getting accumulated in the human. So they're feeling full even before the food comes into the gut. So here we could quantify pre-ingested satiation in humans. Then we asked where the GLP-1 drugs work in the brain. So we got some brain slices uh, from our lab from our institute, and we looked into the hypothalamus, and we could see that there were a lot of GLP-1 receptors in the dorsal medial hypothalamus. So we could label these receptors with immunohistochemistry, and then we could also see that in mice, um, there were GLP-1 receptors in their dorsal medial hypothalamus. So we decided to investigate that the GLP-1, DME GLP-1 receptors in the dorsal medial hypothalamus. So what we did was we first wanted to see the function of these neurons. So the technique we use to see the function of these neurons is a technique called optogenetics. So what this optogenetics does is we deliver a virus into a specific brain region to a specific neuron, and when the virus goes inside the neuron, they make the neurons produce a receptor that can be stimulated by light. And then when the receptors are stimulated by light, they can activate or inhibit a specific neuron whenever we deliver a light. So using these optogenetics, we can control the timing of activation or inhibition of a specific neuron. So using this, we asked if the DME-GLP receptor neurons are causing satiation. So we first inhibited the DME-GLP receptor neurons, and we thought that if these neurons are satiation neurons, inhibiting these neurons would inhibit feeling of fullness, so the mice would have a prolonged bout duration of eating. And as you can see here, you can see that the mice eats more. So here we could see that the DME-GLP receptor neurons are necessary for satiation. And then we asked if the DME-GLP receptor neurons are sufficient to cause satiation. So what we did here was we specifically activated these neurons when the mice started eating in a closed loop manner. So when we did this, you can see here that the mice stop eating right away when the DME-GLP receptor neurons are activated. So here we can see that the DME-GLP receptor neurons are sufficient for satiation. Then we went to see what kind of valence, what kind of feeling activating these neurons would cause. So we thought that because when we feel full, we don't like the feeling and we stop eating because our belly feels so full. So we thought it would produce a negative valence. So what we did here was we activated the neurons each time a mouse went into this specific region of a chamber. So then we can see if the mice likes this region or dislikes this region every time these neurons are activated. And what we could see was the mice changed preference each time the GLP-1 receptor neurons were activated. And we could change the preference of these mouse in a very short amount of time, about 10 minutes. But we didn't see any change in valence when these neurons were inhibited. So to sum up, we could see that through optogenetics, the DME-GLP receptor neurons are causing satiation, but this doesn't show that these DME-GLP receptor neurons are really activated during the pre-ingestive phase, really before eating, because we're just artificially manipulating them. So to look into the natural dynamics of these neurons, we did a technique called fiber photometry. So what we do here is we inject the virus into a wanted region into a specific neuron. So when this virus goes inside the neuron, they make the neurons produce a protein called GCAM, and when the neurons are activated, these GCAM proteins emanate light. So they're correlated with the neuronal activity. Then we can collect these lights by inserting a cannula in the mouse brain, and then we can record them using a machine. So after we made this specific mice, we made the mice do this maze test where the mice have to stay in the shelter and then they have to travel this a long maze in order to get to food to consume them. So this lets us have a sufficient amount of time to record neural activity during the pre-ingestive period. So what we could see was when this is the first time the mice are introduced to the chamber. So when they didn't know about the food at the end of the chamber, we could see that there was no neuronal activity during the pre-ingestive phase. And when the mice met the food, that's when the neurons got started to activate it. But when the mice were conditioned, meaning that when the mice knew about the food at the end of the maze, the neuronal activity started to increase just from the mice coming out of the shelter and starting to travel to the end of the maze. So here we could see that the DME-GLP receptor neurons are activated in this pre-ingestive phase. Then we wanted to see if these neurons encode a more cognitive situation. So we did a T-maze test where we conditioned mice of the food at the goal site. And we recorded neural activity before we conditioned the mice, after the mice learned about this food, and after the mice forgot about the food. So what we could see here was that when the mice chose the non-goal site, we couldn't see any change in neuronal activity. But when when the mice chose the goal site, we could see that the neuronal activity increased after the mice were conditioned. And then, and when the mice forgot about it, you can see that the neuronal activity was diminished. And we could also see that the mice behavior change as we condition the mouse. Then, so here we could see that the DME-GLP receptor neurons are encoding a pre-ingestive satiation. Then we wanted to look into how the individual neurons of the DME-GLP receptor neurons are encoding pre-ingestive satiation. Because the result that we just saw before is all a bulk activity. We're recording all the neurons at the same time. So what we did was we implanted a miniscope into a mouse, into the DMH, and recorded hundreds of neurons that can make a light from the GCamp, and we let them do this maze test again. So we recorded about over 100 neurons, and we used a machine learning technique called choice probability, which can help differentiate what kind of behavior a neuron is more likely to prefer. So we got two neurons, the pre-ingestion neurons and the ingestion neurons. And you can see that the pre-ingestion neurons are activated during the pre-ingestive phase before the mice start eating. But after the ingestion starts, the pre-ingestion neurons deactivate, and the ingestion neurons, they activate. And as soon as the ingestion ends, the ingestion neurons deactivate and the pre-ingestion neurons have a rebound. And what was more interesting is during the no ingestion trial. So in this trial, the mice look at the food, but they know it's there, they even touch it, but they don't consume the food. And what happens is the pre-ingestion neurons, they activate during the pre-ingestive phase, but the ingestion neurons, they don't activate at all. So what this means is that the ingestion neurons, they are actually being activated to the consumption behavior itself to preemptively activate to prepare for the future meal termination behavior. Then we wanted to ask, what will the GLP-1 drugs to the DME GLP receptor neuron? So we recorded neural activity using the fiber photometry, and we injected saline or GLP-1 drugs into the mice while the mice were doing the maze test. So what we could see was that the injecting the GLP-1 drugs to the mice didn't make the overall activity increase. So we don't see the GLP-1 receptor neurons being activated high getting high activation all the time. But when we looked into the behavioral specific phases here, the eating phases, we could see that when the mice started doing feeding behavior, like getting food information or starting to go out search for food, or when they started ingestion, you could see that these GLP-1 drugs make these neurons sensitive to the food information that these mice are experiencing. So these neurons are getting potentiated when the GLP-1 drugs are getting delivered to them. So we think this is similar to the results that we got in humans. So when the humans are treated with the GLP-1 drugs, you can see that they have a heightened sense of satiation when the food information is delivered to to the human. So, and then we finally wanted to see the where the DME GLP-1 receptor neurons project to, what kind of circuit they have in the brain. So we looked into the very famous hunger HRP neurons, because when we start getting full, we feel less hungry. So we thought it would be obvious to look into the one of the most famous hunger neurons in the brain, the HRP neurons. And we did this with collaboration from the UT Southwestern Kevin Williams lab. So what we did here was we took a brain slice of the brain, and then we did electrophysiology recordings to see the circuit activity. So we first activated the DME receptor neurons, and then recorded the neural activity of the hunger HRP neurons, and we could see that activating the DME receptor neurons could inhibit the hunger HRP neurons. Then we wanted to see if the drug itself is required for the activation of the DME GLP receptor neurons. So we first wanted to see if the drugs have a direct effect on the DME GLP receptor neurons. So we blocked the synaptic connectivity in this region, because the GLP-1 could bind to other GLP-1 receptor neurons in a part of the brain, and then they could connect to the DME GLP receptor neurons. So when we blocked all the synaptic connections, and then when we treated the drugs, we could see that the DME GLP-1 receptor neurons were activated. So we know that this is a direct effect. And then when we knocked out the GLP-1 receptor neurons in the DMH, we could see that blocking the synaptic connectivity and then delivering the GLP-1 drugs had no effect on these neurons. So here we know that the GLP-1 receptor neurons are directly activated by the GLP-1 drugs in ex vivo. So then we wanted to see if manipulating the circuit could change the action potential activity in this circuit. So we inhibited the DME GLP receptor neurons while delivering the drugs, and we could see that the inhibition of these neurons could attenuate the increased neuronal activity of the DME GLP receptor neurons, or the decrease of the HRP neurons. And then we wanted to see if the DME GLP if the circuit is required for the activity effect of these drugs. So we inhibited the GLP receptor neurons or activated the HRP neurons. And when we did this, we could see that manipulating the circuit could attenuate the effect of the GLP-1 drugs. So to summarize, we could see that the DME GLP receptor neurons is connected to the hunger HRP neurons to cause pre-ingestive satiation. And then when the GLP-1 drugs bind to the DME GLP receptor neurons, they make these neurons more sensitive to the food information that is getting delivered. And we could reveal this by a translational study from humans and mice. So I'd like to give thanks to all our lab members, and especially Junseok Park. So he was one of the first. He was the one of first to get the data to start up the project, and He also did a lot of the clinical studies. And also, I give thanks to our collaborators, Kevin Williams and Eun-sang Wang. So, Eun-sang is now in the job market. He might be coming to Korea. So, if you guys maybe see him, then maybe you can say hi. And this is a picture of our first international collaboration meeting. So, this is us 8 o'clock in the morning, and this is them, maybe I think like 8 o'clock in the night. And thank you all for your attention.